This lesson is an introduction for Chapter 25, where we're going to be talking about the origin and diversification of eukaryotes. The first representatives of the domain Eukarya, or the eukaryotes, are thought to have arose about 1.8 billion years ago through the process of endosymbiosis, and we'll be revisiting that topic on the next few slides. We have also talked about it previously when we were talking about the prokaryotes. What is it that differentiates a eukaryote from a prokaryote? The main thing that you should remember from Biology 1 is that eukaryotes have a nucleus that contains their DNA, whereas prokaryotes do not have a nucleus. Eukaryotes also have membrane-bound organelles, such as mitochondria, chloroplasts, endoplasmic reticulum, and others. And those organelles are the sites of particular cell functions. They're like tiny organs that have very specific jobs in the operation of a cell. The earliest eukaryotes were unicellular, but now there are more diverse forms of eukaryotes, including both unicellular and multicellular forms. Something else that differentiates eukaryotes from prokaryotes is a well-developed cytoskeleton that enables eukaryotic cells to have different forms, change shape, such as shown here in this scanning electron micrograph of a ciliate didymium engulfing a paramecium through phagocytosis. The only way that the ciliate is able to engulf that paramecium, which looks kind of large for its own body, is by changing its own shape to surround it and take it into its body. So that is something that was not seen before with the prokaryotes that we now see in some eukaryotes. As you might recall, we talked previously about the evolutionary similarities between the three domains, the eukarya, the archaea, and the bacteria, and that the eukarya is more closely related to one domain than the other. In fact, let me pull up that diagram real quick as a quick recap. Okay, here's that phylogenetic tree of the three domains. So as we have already discussed, the eukarya are more similar to archaea, their sister clades, than eukarya or archaea are to bacteria, which would be considered the outgroup of this phylogenetic tree. However, all of these domains have a common ancestor, the ancestral cell of all life that came later. So when we look at the slide I was just showing, we can see that the eukarya have inherited some properties from both that are seen in both the archaea and the bacteria, which we can presume they all have because of that common ancestor. Okay, so now let's take a better look at this diagram, this table rather, showing evidence of the eukarya having both archaeal and bacterial origins. Now the majority do seem to have archaeal source, such as the enzymes involved in DNA replication, transcription, and translation, all part of the mechanisms that the cells can make copies of themselves or used to make proteins from the DNA, that machinery. Also, in terms of how cells can make copies of themselves through cell division, the apparatus is similar to archaeal in the eukarya. For the endoplasmic reticulum, there's both archaeal and bacterial source materials. But for things that are just from the bacterial origins would be the mitochondrion and the metabolic genes. And if you recall, we were just mentioning this again, that the eukaryotes were thought to have come through the process of endosymbiosis. So as we're looking at these features and where they originated from, we have to bear in mind that theory about endosymbiosis to bring some context to this. So if you recall, again, this is also a review, this theory tells us that the earliest eukaryotes would have engulfed some prokaryotes that were free living, and instead of digesting them as food, they retained them in their cytoplasm, and they served a benefit to the eukaryote that they were within. that would be a mutualistic relationship when both parties are benefiting. So it's showing here two different possible scenarios here. One would be on the left, the ancestral heterotrophic eukaryote that came from an ancestral prokaryote that was engulfed by a eukaryotic cell, and then that became the mitochondrion of the cell, of the eukaryotic cell. And the other scenario shows there already would be a mitochondrion, and that then, so that the first thing that would have been engulfed would have had to be the mitochondrion, and there was already that eukaryotic cell with the mitochondrion, which then engulfed a photosynthetic bacterium, which would be cyanobacteria, which now is what we call a plastid, a photosynthetic organelle. It could be a chloroplast, or it could be a different kind of pigment molecule, pigment-containing organelle. So these are the descriptions that accompany that visual that we just looked at about the endosymbiont theory. So if you need to take a moment to recap that, you can pause and read this alongside the diagram. But I'm going to go head on as we further explore the evidence for endosymbiont theory. Of course, nobody was there to see that happening, so it's all speculation and theory, but there is evidence that supports that theory. One piece of evidence is the composition of the inner membranes of the mitochondria and the plastids, they have double membrane. They have an inner membrane and an outer membrane. The inner membrane is similar to the plasma membrane of a prokaryote, in contrast to the outer membrane, which is more similar to a eukaryotic plasma membrane. So if you look back at the diagram, notice that the mitochondrion, which again would have been a free-living prokaryote, had its own membrane, and then that would be the inner membrane of the mitochondrion now in the cell. And the outer membrane would have been derived from the cell membrane of the eukaryote that engulfed it, forming its own little, in a way like a food vacuole, but it didn't digest it. It just retained that outer membrane, which came from the cell membrane of the eukaryote. Another piece of evidence for the endosymbiont origin of mitochondria and plastids like chloroplasts is how they can divide similar to prokaryotes, which go through binary fission. Chloroplasts, other plastids, and mitochondria can also divide and duplicate inside of the eukaryotes like that. In addition, plastids and mitochondria have DNA. In fact, eukaryotes, the majority of their DNA is in the nucleus, but there is some DNA in the mitochondria and the plastids, and that is similar in structure to that of prokaryotes. And having their own DNA means that that DNA can also be transcribed and translated within those structures, within the mitochondria, within the chloroplasts, suggesting that with all the machinery there already, that those organelles themselves could have been a free-living, single-celled organism before they became part of a larger eukaryote. In addition, well, in order to make proteins from DNA, they have to have ribosomes. So ribosomes inside the mitochondria and the plastids are similar to prokaryotic ribosomes, and not as similar as they are to eukaryotic ribosomes in terms of their size and sensitivity to things like antibiotics. This, of course, has been studied by scientists to come to these conclusions.